Now that is an example of something that activates your 3-neuron arc. Now let's go into more depth on how it works.
So this is the design of a neuron, and we're going to start with the axon. And the axon is that line right there, and I'm going to draw it for you. It's that thread-like line, and is the part of the nerve cell along which impulses are conducted and are formed from the cell body to other cells. And then we're going to go to the axon terminal, which is up here. It's that right there. That's the axon terminal.
And this is the part of the nerve cell that makes synaptic connections with another nerve cell or effector cell. And the axon hillock is the site of summation for incoming information and is a specialized part of the cell body of a neuron that connects to the axon of the cell. And then the myelin sheath allows electrical impulses to transmit quickly and efficiently along the nerve cells.
We're going to go to synapse, and this is synapse right here. It's really tiny, but there's a bigger picture right there.
And a synapse is the junction between two nerve cells consisting of a minute gap across which impulses pass by diffusion of a neurotransmitter.
So now we're to the cell body, and it's like the area that surrounds the nucleus, the area around it, and it connects the dendrites to the dendrites, which bring information to the neuron, and the axon, which sends information to other neurons. Then we're going to go to dendrites, which are just a little below. They're right there, and they're like the little tree-like structures right there. And they are designed to receive communications from other cells. They resemble a tree-like structure, forming projections that become stimulated by other neurons and connect the electrochemical charge to the cell body. Then we're going to go to the nodes of Ranvier. And those are right there. They're like the space between the myelin sheath. Put in a different color. Right there. And like right there too. And these allow the generation of fast electrical impulses along the axon. And then we're going to go to synaptic vesicles. And they're up there right here. And they're like those circles right there. And these play a central role in neurotransmission as the transmitter storing and releasing organelles of the nerve terminal. So then we have the sodium channels, which mediate fast depolarization and conduct electrical impulses throughout the nerve, muscle, and heart. And it's in pink circled, that's the sodium channels. And then we're going to do the voltage-gated calcium channels, which is what I circled in yellow, which is on the synapse actually. And those mediate calcium in response to membrane depolarization and regulate intracellular processes such as contraction, secretion, neurotransmission, and gene expression in many different cells. Then we're going to go to the nucleus, which is up here, and I highlighted it in light blue, and it's the center of the cell, and it contains genetic material in the form of chromosomes, and it's like all the DNA of your cell. And then we're going to go to the mitochondria, which is the dark blue surrounding the nucleus, and it is the powerhouse of the cell. So first we have the voltage-gated sodium channel, which is an integral membrane proteins that change conformation in response to depolarization of the membrane potential and open a transmembrane pore to conduct sodium ions inward to initiate and propagate action potentials. Then we're going to go to the synaptic cleft, which is the space between the synapse and the neuron, and it's highlighted in yellow. It's that line in yellow, and it is the space that separates two neurons, and it forms the junction between two or more neurons and helps nerve impulses pass from one neuron to another. Then we're going to go to the protein receptors, receptor proteins, and it's circled in green, and that is molecules that inside the target cell or on its surface to that receive a chemical signal. Then we're going to go to the cell membrane, which is up here, and the cell membrane is like the outside, and it completely separates the cell from its external environment, and it keeps everything out that's supposed to be out and everything in that's supposed to be in. Then we're going to go to the neurotransmitters, which I circled in blue right there, and it is a chemical substance that is released at the end of a nerve fiber by the arrival of a nerve impulse and is diffused across a synapse or junction, causes transfer of the impulse to another nerve fiber, muscle fiber, or some other structure. So now we're going to talk about the connection of the motor neuron, interneuron, and sensory neuron in the 3-neuron arc. So it is like a reflex. So when your body gets a sense of something that is pain or a reaction of some sort, the reflex acts before your brain even processes what happens due to the 3-neuron arc. And so here's an example. So say you touch a needle or a pin, and which is a stimuli, it touches the receptor cells, which then go to your sensory neuron, which go up to your spinal cord, then into your relay neuron or the interneuron to the motor neuron, which is like the effector, and it makes your muscle or something contract to move away from that needle. So now we're going to talk about the electrical graph, and first it starts at its resting state, which has to be at -70, which is the resting potential. Then in order to generate an action potential, the stimulus must be strong enough to bring the intracellular neuron to a critical threshold of -55, which is what it's going to be next. And then once the threshold is exceeded, the sodium channels open and the sodium moves inside intracellular polarity, shifts from a -70 towards a positive state of positive 35 to be exact, and this is known as depolarization. And then as the action potential reaches its peak, the sodium channels close and the interior of the neuron cell is now positive, and as those sodium channels close, the potassium channels open, and the potassium moves outside the cell, creating a negative polarity known as repolarization, and then potassium continues to escape the neuron and goes into hyperpolarization, and it dips below the resting membrane potential, and then this cannot be stimulated, and it is known as the refractory period, and then the potassium gates close, and it is restored back to its resting potential. So the 3 neurons that there are in this are the motor neuron, interneuron, and sensory neuron. And the motor neuron have long axons and transmit nerve impulses from the central nervous system to effectors. And then the interneuron, which is in the middle, or the relay neuron, are usually much smaller cells with many interconnections. And then the sensory neuron have long axons and transmit nerve impulses as well from sensory receptors all over body to the central nervous system. And then the neuron communication with the synapse, which is really important to this, is first the information of the axon needs to get to the cell body or the dendrite, and this happens with the use of synapses. And first the action potential is moved down the axon terminal, which triggers the vesicles to move down the neuron, then cause the release of neurotransmitters to the neuron into the receptors on the other side, then causing the action potential to move down the other neuron. So those green bubbles, the vesicles, are moving down because of the action potential to then release the neurotransmitters into the cleft to connect to the receptors to then move to the other neuron through, and then the action potential moving them out, and then those neurotransmitters then disappear after that from the cleft.